Hello everyone, I'm Anas Al-Ubaidi, and here we have the first lecture or first session in chapter 12 of the certification course for Advanced Medical Science Liaison and Medical Monitor program by CCRPS. And here, chapter 12 is about the data management and risk assessment. And the first topic or first lecture is ensuring data accuracy, reliability, and handling missing data. Introduction. Data accuracy, reliability, and the effective handling of missing data form the foundation of high-quality clinical research. So, in order to maintain one of the essentials, one of the requirements, in order to maintain high-quality clinical research, is considered as foundation is to have data accuracy reliability. And in addition to that, the effective handling of missing data. These principles ensure the integrity and interpretability of results, supporting valid scientific conclusions and regulatory compliance. So, data management in general within clinical research is the foundation of high quality clinical research. Clinical trial data by its nature must precisely represent the study's population and intervention outcomes. Any deviations or inaccuracies in data collection, processing, or assessment compromise not only the trial's validity but also patient safety, participant safety, and the broader credibility of the research. Data reliability ensures that results are consistent, reproducible, and free from external biases, errors introduced by equipment, human processes, or variability across sites. Meanwhile, managing missing data effectively now requires sophisticated evidence-based strategies that maintain transparency and minimize the risk of bias, of errors. For advanced physician medical monitors, for MMs, these data stewardship tasks are more critical than ever in an era of complex protocols and decentralized trials. With the integration of advanced tools, methods, and concepts into clinical research, they must oversee data accuracy and reliability throughout the trial life cycle while proactively identifying and addressing gaps in the dataset. Key points. Ensuring data accuracy. So, definition and importance. Data accuracy measures how the accuracy of the data measures how well recorded data represents the actual values observed in clinical research. Accuracy is a non-negotiable component, as even minor errors, minor bias in data recording or processing can cascade into flawed conclusions, regulatory penalties, and/or compromised patient participant safety. Techniques for ensuring accuracy. Standardized data collection procedures. Uniformity in data collection reduces variability and errors. Example: blood pressure measurements are consistently taken using calibrated devices, ensuring all trials use standardized units and techniques. Automated data validation systems integrated into EDC, electronic data capture systems. These tools flag incomplete or outliers entries. Example: if entered platelet counts fall outside the biological plausible range, the system auto-generates an query for site clarification, meaning if the wrong data or inaccurate data was entered in a place in different shape how it's supposed to be entered, In that particular section in EDC, or electronic data capture system, will generate something called auto queries, or automatic queries, or automated queries, systematic automated queries, and this is called automated data validation systems. Source data verification, SDV. Cross-referencing case report forms, CRFs, with original medical records ensures the authenticity of trial data. Example: direct comparison of patient eligibility data from the clinic's database with CRFs submitted to sponsors. Comprehensive staff training. Conduct regular training to ensure consistency in understanding and applying trial protocols. Example: a dedicated session for capturing adverse event data ensures alignment across sites. Robust data backup mechanisms. Systemic failures can lead to data losses, making secure backups vital. Example: encrypting all data transfers to ensure retention during EDC software upgrades. And here we have a nice summarized diagram, or summarizing diagram, showing or explaining about the ensuring or insurance of data accuracy in clinical research. As we have here, SDV, source data verification, automated data validation, which is a particular one of the most famous kind of it is the systematic auto queries, standardized data collection, and data accuracy. Strategies for data reliability. Definition: reliability refers to the consistency and replicability of measurements or data points collected from the same conditions over time. Unreliable data can skew trial results and jeopardize their acceptance by regulators and peers. Key strategies as SOP, site or standardized operating procedure-driven procedures, detailed workflows for data entry, sample collection, and reporting ensure consistency. Example as SOPs, site or standardized operating procedures define how tumor sizes must be measured, specifying the imaging equipment and measurement techniques. Inter-rater reliability checks as consistency tests among multiple investigators or principal investigators or raters reduce observer bias errors. Example: a study involving liver ultrasound measurements checks alignment between 3 radiologists to ensure consistent scoring. Calibration of instruments. Equipment such as centrifuges or glucose monitors must be calibrated regularly to avoid data drifts. For example, bi-monthly recalibration of lab equipment in multinational trials ensures accuracy across sites. And this is just an example, because the majority is annual yearly recalibration of the instruments at the research site facility. Routine data audits. Implement periodic audits throughout the trial to flag recurring errors or irregularities. Example: reviewing a random selection of 15% of primary endpoint data entries per quarter identifies transcription discrepancies. Training for consistency. Refresher workshops on measurement methods and eligibility reviews minimize inter-trial variability. Example: standardized training for emergency response procedures during serious adverse events, SAEs. Continuous monitoring. Centralized monitoring tools now provide real-time alerts on data reliability deviations. Example: alerts are triggered if data patterns suggest significant manual transcription errors from one specific trial site. Here is a summarizing diagram showing the strategies for data reliability, calibration of instruments, SOP-driven procedures, inter-rater reliability checks. Handling missing data. Definition and possible bias, errors. Missing data occurs when expected data points are unavailable, introducing gaps that could skew analysis and create misleading outcomes. Types of missingness must be understood to apply the correct interventions effectively. Types of missing data: missing completely at random, MCAR, entirely unrelated to the study variables or conditions. Example, lab samples are accidentally destroyed in transit and no other identifiable factor influences the loss. Missing at random, MAR, associated with observed data but not unobserved data itself. Example, elderly patients are more likely to drop out of data collection due to fatigue during extended follow-up periods. Missing not a random, MNAR, missing data is related to unobserved values. Example, patients with severe side effects intentionally withdraw without clarifying their reasons. Techniques for managing missing data. Data prevention measures. Thorough trial participant communication emphasizing attendance and compliance. Example, automated reminders via SMS for follow-up visits reduce dropouts. Multiple imputation approaches. This method generates multiple datasets by replacing missing values with plausible estimates to reduce bias. Example, missing blood sugar levels are imputed using baseline BMI, body mass index, and comorbidity data trends. List-wise exclusion, removing datasets with missing primary endpoint values, though often a last resort due to potential data loss. Example, participants without any follow-up data for primary outcomes are excluded from analysis. Sensitivity analysis, testing trial outcomes under various assumptions around missing data. Example, running analysis with both imputed and excluded datasets to ensure consistent endpoint conclusions. Machine learning algorithms, advanced prediction models compensate for missing values. Example, a neural network predicts dropout patterns and suggests flexible rescheduling to reduce participant loss. And here is a summarizing diagram showing how to handle missing data. As here we have the MCAR, MNAR, and MAR. As MCAR, unrelated to study variables, often due to random errors. MNAR, related to unobserved data, often leading to biased results. MAR, related to observed data, such as demographic factors. Benefits: enhanced data integrity. High quality, accurate data strengthens the foundation for robust regulatory approvals. Example, early detection of serious adverse event SAE trends reduces post-trial adjustments. Reliability across multi-site trials, effective strategies ensure uniformity across global sites, building credibility and validity into outcomes. Reduced operational costs by minimizing errors and missing data. Trials avoid costly or corrective procedures. So here a diagram about data integrity and trial efficiency. We have the data integrity, reliability, and cost reduction. Challenges and recent advances. As challenges large volumes of data, multinational trials generate unprecedented data points, complicating validations. Solution scalable dashboards for automated data screening. Staff resource gaps, low staff to data ratios hinder thorough oversight. Solution is hybrid approaches balancing manual reviews with AI, artificial intelligence algorithms. Advances here: AI, artificial intelligence, and predictive analytics. Real-time trend analysis helps forecast data risks, adapting oversight strategies based on anomalies. Example, Algorithms predict dropout likelihood based on patient cohort behavior. Decentralized monitoring with cloud technology as EDC electronic data capture platforms enable live data sharing, reducing reliance on local site tools. Example, monitors verify monitors verify recruitment data through cloud dashboards weekly. As current advancements in structured workflow flows provide unparalleled capabilities in safeguarding data quality. These systems, deployed effectively by advanced physician medical monitors (MMs), ensure trials are both compliant and scientifically rigorous while maintaining participant-centered approaches. Here's a good diagram. Explains the AI, artificial intelligence enhances data oversight in trials, as we have AI analytics, improved compliance, enhanced data quality, increased efficiency. Summary tables, as the following tables summarizing the or our summary for the clarifications and the um, what have been or has been explained. So table 1, key techniques for ensuring data accuracy. The technique, description, purpose, example, challenge. Techniques, we have standardized collection, automated validation, training and education, source data verification, SDV, and real-time data capture. Another table about strategies for ensuring data reliability. We have strategy, description, benefit, example, and challenge. Strategies, reproducible procedures, routine data audits, instrument calibration, inter-rater checks, sensitivity, and sensitivity assessments. Types of missing data and handling techniques. The type, definition, key example, technique, and challenge. As types, we have the MCAR, MAR, MNAR, imputation methods, and prevention strategies. Challenges and their solutions or resolutions in data management. You have the challenge, impact, solution, implementation example. Challenges are large trial datasets, limited resources, technological gaps, high participant variability, time-sensitive errors. Recent advances or advancements in handling data issues. The advancement, description, benefit, use case, and example. Advancements, we have the cloud data systems, predictive analytics, AI, artificial intelligence for imputation, virtual decentralized tools, and digital dashboards. Application table, we have the scenarios. For each scenario, the key principle that was applied, key actions, timeline, what to watch out for. As scenarios here, we have errors in blood pressure measurements due to varying equipment, missing glucose readings in a diabetes trial, frequent transcription errors in case report form entries, dropout of elderly patients in a cardiovascular trial, measurements, inconsistencies in radiology imaging. Continuing with scenarios, delayed adverse events reports due to staff turnover, loss of lab samples during transportation, variability in tumor imaging results across multiple locations, unclear patterns, and serious adverse event SAE reporting delays. And then we have, as a last scenario here, low quality data flagged during data integrity audit. We will revise some MCQs, multiple choice questions, as question number 1. What is the definition of data accuracy in clinical trials? Is it A, the consistency of data collection methods over time and across sites? Is it B, the degree to which data correctly represents real-world values it models? C, the process of filling in gaps for missing data points. D, the probability of missing data occurring at random. So it is B, the degree to which data correctly represents real-world values it models. Data accuracy ensures the data aligns with the actual events, measurements, or conditions it is intended to depict, which is essential for valid trial outcomes. Question 2. Which method can enhance the reliability of data in clinical research? Is it ignoring data inconsistencies during audits? A. B. Standardizing operating procedures across all sites. C. Removing missing data points without addressing their impact. D. Using uncalibrated measurement tools to save time. So it is B. Standardizing operating procedures, SOPs, across all sites. As for sure, it's not ignoring data inconsistencies. For sure, not removing missing data points. For sure, not using uncalibrated measurement tools. So it is B. Standardized or standard operating procedures (SOP) ensure that data collection and analysis remain consistent, fostering reliability across different sites and personnel. Number three, what is a defining characteristic of missing, not a random MNAR data? Is it A, data is missing due to hardware or technical issues? Is it B, missingness is unrelated to both the observed and the unobserved data? C, missingness is systematically related to unobserved data, or D, as data is unrelated to observed data set characteristics. So it is C, missingness is systematically related to unobserved data, as MNAR occurs when the missingness is connected to factors not present in datasets, such as a participant withdrawing due to adverse effects unknown to the researcher. Number four, what technique prevents data loss during clinical studies? Listwise, A, listwise deletion of all participants with missing outcomes. B, sending reminders to participants for follow-up visits. C, ignoring missing data to speed up analysis. D, using no imputation methods to fill gaps. So it is B, as sending reminders to participants for follow-up visits. Example, reminders reduce the likelihood of dropouts and incomplete datasets, preventing data loss and improving overall data completeness. And five, how does multiple imputation address the issue of missing data? It removes all records with missing data for accurate results. B, it fills in missing points with predicted values across plausible datasets. C, it replaces missing data with a single outcome variable's mean. Or D, it avoids analyzing missing data cases completely. So it is B, it fills in missing points with the predictive values across plausible data sets, as multiple imputation estimates missing data using related variable and generates multiple possible data sets for robust and unbiased statistical analysis. And here we come to end of this video, to end of this session or lecture. Thank you very much and goodbye.